Hello and welcome to Sam Biology class. Friends, today we shall discuss about the processing of pre-mRNA. Before that, I request all of my friends, please subscribe my YouTube channel. Your subscriptions are a lot to me. Pre-mRNA or precursor mRNA is the primary transcript of a protein coding gene. It is the mRNA which has not undergone any modification after transcription. In eukaryotes, the pre-mRNA contains both protein coding exons and non-coding introns. Therefore, in order to produce fully mature and functional mRNA, the primary transcript in eukaryotes undergo extensive modifications. All these modifications together constitute post-transcriptional modifications or processing. In bacterial cells, transcription and translation takes place simultaneously. Because transcription and translation are coupled, there is a little opportunity for the bacterial mRNA to be modified before protein synthesis. In contrast, transcription and translation are separated in both time and space in eukaryotic cells. Transcription takes place in the nucleus, whereas translation in the cytoplasm. This separation provides an opportunity for the eukaryotic mRNA to be modified before it is translated. Indeed, the eukaryotic mRNA is extensively altered after transcription. During processing of pre-mRNA, changes are made to the 3' as well as the 5' end of the primary transcript. In addition to this, its protein coding segment is also altered. In our discussion, we will focus on 3 important post-transcriptional modifications that the eukaryotic pre-mRNA undergoes during processing. First one is capping, second one is polyadenylation, and the third one is splicing. What is capping? Capping is the addition of a structure called a 5' cap. The cap consists of an extra nucleotide at the 5' end and methyl groups on the 7th position of the base of the newly added nucleotide and on the carbon to hydroxyl group of the sugar of one or more nucleotide at the 5' end. Capping takes place rapidly after initiation of transcription. This figure illustrates the pathway of mRNA capping in which several different enzymes are involved. Capping requires the sequential action of 3 different enzymes such as RNA triphosphatase, guanylyl transferase and methyl transferase. Normally, all RNAs have 3 phosphate groups at the 5' end. These phosphate groups are designated alpha, beta and gamma. mRNA capping begins with the removal of the gamma phosphate by the action of enzyme RNA triphosphatase. After removal of gamma phosphate, a guanine nucleotide is added to the 5' end by an unusual 5'5' bond, which is different from the normal 5'3" bond seen in nucleic acids. This reaction is catalyzed by the enzyme guanylyl transferase and is coupled to the release of an inorganic pyrophosphate. Next step is methylation, in which methyl groups are added. These steps are catalyzed by the enzyme methyltransferase. The first methyl group is added to the 7th position of the base of the terminal guanine nucleotide, thereby making 7-methylguanine. This is followed by the addition of methyl groups to the carbon to hydroxyl group of the sugar of 2nd and 3rd nucleotide. Rarely, the base of the 2nd and 3rd nucleotide may also be methylated. The initial enzyme in this pathway is associated with RNA polymerase II. Neither RNA polymerase I nor RNA polymerase III are associated with this enzyme. As a result of this, ribosomal RNA, transfer RNA, and some small nuclear RNAs are never kept. The second type of modification to the eukaryotic pre-mRNA is the addition of 50 to 250 or more adenine nucleotides at the 3' end, forming a poly-A tail. These nucleotides are not encoded in the DNA but are added after transcription. Many eukaryotic genes transcribed by RNA polymerase II are transcribed well beyond the end of the coding sequence. Most of this extra material is cleaved and then adenine nucleotides are added to the new 3' end by a process known as polyadenylation. Processing of the 3' end of pre-mRNA requires the presence of specific sequences known as polyadenylation signals. One of the sequences is AAU-AAA, which is located 11 to 30 nucleotides upstream of the site where cleavage will take place. This sequence determines the point at which cleavage will take place. The other sequence is rich in either uracil or uracil and guanine. This sequence is located downstream of the cleavage site. So at this point, cleavage takes place and then this extra material is removed. After this, adenine nucleotides are added to the new 3' end by an enzyme poly-A polymerase or polyadenylate polymerase. So in this way, a long poly-A tail is added to the 3' end of the eukaryotic mRNA. Poly-A tail confers stability on the mRNA. It increases the time during which the mRNA remains intact and becomes available for transcription. Many mRNAs, microRNAs, and small nuclear RNAs are polyadenylated at the 3' end. The third major type of modification of eukaryotic pre-mRNA is the removal of introns by splicing. Splicing takes place in the nucleus before RNAs move to the cytoplasm. Splicing requires the presence of 3 sequences within an intron. One end of the intron is referred to as the 5' splice site and the other end is referred to as the 3' splice site. These sites have short consensus sequence. Most introns in pre-mRNA begins with GU and ends with AG. A third sequence, which is also crucial for splicing, is present at the branch point, which is an adenine nucleotide. The branch point is located about 18 to 40 nucleotides upstream of the 3' splice site. The sequence around the branch point had no strong consensus. Splicing takes place within a large structure called spliceosome. It consists of 5 RNA molecules. And almost 300 proteins. These RNAs are small nuclear RNA, the length of which varies from 107 to 210 nucleotides. These RNA components associate with different proteins to form small nuclear ribonucleoprotein particles. Each snRNP consists of one molecule of snRNA and multiple proteins. The spliceosome is composed of 5 small nuclear ribonucleoprotein particles, which are designated as U1, U2, U4, U5 and U6. Some proteins are not associated with an snRNA. This figure illustrates the pathway of splicing. Before splicing takes place, an intron lies between an upstream exon and a downstream exon. Splicing takes place in two distinct steps. In first step, the pre-mRNA is cut at the 5' splice site. This frees exon 1 from the intron, and the 5' end of the intron is attached to the branch point, as a result of which it folds upon itself, that result in the formation of a structure called a lariat. In this reaction, the guanine nucleotide present in the consensus sequence of the 5' splice site bonds with the adenine nucleotide of the branch point through a transesterification reaction. So in a single reaction step, both 5' cleavage and lariat formation occurs. The result is that the 5' phosphate group of the guanine nucleotide is now attached to the carbon to hydroxyl group of adenine nucleotide of the branch point. In the second step of splicing, a cut is made at the 3' splice site, and simultaneously the 3' end of exon 1 becomes spliced or covalently attached to the 5' end of exon 2. The intron is released as a lariat, which is then converted into a linear form by specific debranching enzyme in the nucleus. The linear intron is then rapidly degraded by nuclear enzymes. After removal of all introns, the mature mRNA, which has a 5' cap and a 3' poly tail, moves to the cytoplasm for translation. In next video, we shall discuss how various small nuclear ribonucleoprotein particles of the spliceosome participate in the splicing pathway. For more updates, subscribe my YouTube channel, Sam Biology Class.